Dr. Madhav. He's a endowed Distinguished Professor of Biocomplexity, and he's the Executive Director of the Biocomplexity Institute here at the University of Virginia. He's a passionate advocate and practitioner of transdisciplinary team science, and in his 30-year career, he's been involved in a lot of really major projects in transdisciplinary science. He's an amazing researcher with over 500 articles in peer-reviewed journals, conferences, and workshops. He's been involved in mentoring and training throughout his life, including co-advising more than 30 postdoctoral students, 20 MS students, and 15 postdoctoral fellows. We're really excited to have him here today, and he's going to talk to us about work that he's done at Biocomplexity in real-time pandemic science. So Madhav, I'll turn it over to you, and thank you so much for being with us.
Thank you very much, Don. It's a very kind introduction, and really delighted to join you all today and learn a little bit more about the work you all do as well. I remember when I first came to UVA back in 2018 or so, I met Don, and talking to him was very influential in our final decision in coming here. So let me start. I want to thank iThrive organizers, Sandra, Karen, Jennifer, and others who kindly invited me. Also want to thank folks at BI, as well as our collaborators at other universities and centers, as well as sponsors. They are really crucial because we can't do the work without the funds we get. Everything I'm going to speak about is work that is done in a team-like environment, and I'll say a lot more about it. In fact, I think that as time goes by, my contribution to the team science is becoming smaller and smaller, and it'll soon become vanishingly small. But there's just a wonderful set of folks here who have contributed to this work. So let me give you a brief background about what BI is, because some of you might not know about this as much. Of course, Don and Karen and Sandra, you know our institute very well, but I think for others it might be useful. So we have used the term biocomplexity to refer to interactions between living and non-living agents and their ecologies, including humans and their enabling technologies. And we've chosen this broad definition because motivated by the work that Rita Colwell and Simon Levin have done. They were the ones who actually coined this term biocomplexity back in late 80s. And we wanted to include non-living agents as well here, because as time goes by, these software bots and agents are, cars, devices are becoming an integral part of this ecology, and they shape what we do, and they're shaped by us as well. The way we do our work is driven by mission to solve projects for our sponsors, but we end up developing fundamental science and technology solutions in doing that, and I'll say a little bit more about that as well. We also focus on teamwork, and we undertake problems that cannot be solved by a single faculty member or within a narrow boundaries of a discipline. And finally, we require a sort of diversity in terms of intellectual resources, tools, and ideas. We're focused a lot on societal impact problems, and hopefully the talk today will try and give you an example of the kind of work we have done here. So we essentially end up studying very large, massively interacting systems with between 10 to the 4 and 10 to the 16 agents. And I use the word agents fairly generically. It might be humans, and cell phones, it can be humans, cell phones, and bots, it could be cells within a body, and we have studied all those things in different projects. The key aspect is the size is not so small that you can quickly give a very detailed representation, and it's not so big that you can use principles from physics and build easy compartmental models and throw away the irregularities. And that's what we call it mesoscopic systems. They're right in the middle where details matter to a large extent, and they need to be represented. Otherwise, you will not get the systems that we're talking about. And we've largely focused on biosocial systems. We haven't studied, for instance, pure physical systems as much in the group. So the kind of questions we undertake are things like how can you detect and respond to the next pandemic outbreak, or how can you support the development of sustainable social habitats? Those are very large questions. No single person can claim to solve it, and we cannot essentially really address it even within the institute itself. So we need to build large teams. So I wanted to give you a brief history, minute history, because the work started long time back. I was a postdoctoral fellow back at Los Alamos in 1990s, where I started working on this with a project called Transims that Chris Barrett led. That led to work after 9/11 on a program called NESAC that was set up by a new office and new department that the US government established in Department of Homeland Security. And then once we moved to Virginia Tech, we continued to work with the DoD and at UVA on projects related to national security, AI, and pandemic science. And this tells you an arc of these things. The key part here is that most of these projects are fairly large. They're not small projects. And I'll give you an example of Transims. And they last for a long time. And they leave a very interesting and measurable trace of technologies behind, which is what is of interest to us. So just as one example from the very early period that I started my work on this topic, and then I'll come to pandemics, which is how we'll bring to the discussion more current aspects. But Transims was a program, interesting enough, that started back in 1991-92 timeframe and was funded by the Federal Highway Administration and the Environmental Protection Agency. And it was done at Los Alamos, where I was, and was set up to address important societal problems, assessing traffic flows during congestion, or predicting household behaviors when infrastructure changes happen, assessing social justice issues in proposed changes, and predicting environmental impact, things that are now quite interesting in terms of Clean Air Act. That was a question that they really wanted us to study. So we worked on Transims for a 10-year period. The total amount of funding that the Department of Transportation put on this, including us and later part at IBM and other places, is close to $100 million, just to give you a sense of the size of the project that went on for 10 years. And one of the studies we did at the very end, just as we are moving to tech, was this work that's called the WHATS study, WHATS, the White House Area Transportation Study, that was led by AECOM after we had finished development of the technology. And the question was to see the impact of street closures in and around White House. I think folks who have been going to DC pre 9/11 and post, you've seen that there's a bigger perimeter around White House that you cannot drive close to. And the question was, what sort of traffic and other impacts does it have? And we studied various options like closing down certain streets, building tunnels, extending the metro, having buses, because all the solutions that were proposed had the pros and cons, right? The security was important metric, but travelers' efficiency was important part too. So that was the work that we ended up doing. Really interesting work that the ECOM folks led using the tools we built. And so far, Transims has been used over 60 to 70 different studies now after we finished the project. So I'm going to talk about pandemic science, but let me say a little bit about transdisciplinary team science. Sandra requested me to talk a little bit about that aspect, which I think is really important. So I thank her for asking me to do that. And I've used information from a talk that I gave and a series of notes from Dr. Hall. And NIH has been very interested in this concept for a long time, as you all know. So they define transdisciplinary team science, as I've stated here. It's a process in which a collection of individuals with expertise in different fields come together and synthesize a new body of knowledge using approaches and methods that are specific to their disciplines, but end up developing new ones as well, which is actually a really interesting idea. And they talked a lot about this. And this cartoon, to me, actually does a very good job in explaining at least what Dr. Hall and others felt was transdisciplinary science. So they the analogy was that a unidisciplinary science is a single apple, while multidisciplinary science is akin to a tray of different fruits. To interdisciplinary science, which is like a fruit salad, where the fruits have come together, but yet they maintain their properties. And then transdisciplinary science is really building a shake with all the fruits merged together, and they have lost their individual identity and creating a new idea. I think it's a really nice way to describe, and I thank the authors for thinking about it in this form. And this is really what happens. So, for example, in transportation science, before we did our work, all the work was based on what's called a four-step model. After we finished Transim's project, a new way of modeling transport system has come about and has become the de facto standard of modeling transport science. If you look at any reasonable department and researcher at DOT, they will use Transims-like approach. Now, of course, Transims itself has been changed a lot, but it has produced really fundamental new concepts in the area and changed how transportation systems are viewed in the process. So in our work, we have basically decided to take sort of 5 steps in this process. Work to, step 1 is to have a common goal. Then step 2, which is sustain a stable funding, which is really important, as you can imagine. Third step is to nurture a culture of diversity and respect, which is central to this view of transdisciplinary team science. And then in our work, we have tried to grow these teams bottom up within the institute. We have hired folks who are postdoctoral fellows, graduate students, who have gone on to become very senior scientists within our group. So a couple of important names here: Brian Lewis, who many of you might actually know, done remarkable work, co-led with me a lot of work on pandemic response, actually joined us as a graduate student. In fact, myself, I joined the lab as a postdoctoral fellow and then grew through the ranks. Henning Motwhite is another person who started as a graduate student and is now a faculty member at the UVA. Anil is another person. So the idea is that this sort of brings a sense of culture and a respect for diversity in all forms. And you finally end up working with policymakers and end users and stakeholders to try and solve their problems. There are challenges to doing this. There are conceptual and scientific challenges. The field is being developed as you work together and makes for a challenging set of questions. The cultural variation among team members with different disciplinary backgrounds is another challenge to deal with. Third is management and leadership challenge. You have to try and figure out how teams can work together and in a manner. And the fourth part is really important, which is incentives, recognition, and evaluation. It might seem easy, but I can tell you that this is actually a central portion. And oftentimes, if you allow me to say this, I feel in academic environments, there is too much focus on a single individual or single scientist, and all the evaluations are done with what they have achieved. But I think this view of team science really allows you to understand the collective value that the group brings. And I for one, think that it's an important way to do science. It's not that that's the only way to do it, but I do want to encourage folks on this call to think about doing science that way. It's a different form, and it's equally important, in my opinion, to nurture this form of science because it yields different kinds of systems and tools and ideas. I'm happy to take questions, by the way, in the middle of the talk, so it's not a problem for me, but I don't know what the protocol is here. So let me continue then, but feel free to, I can't see all hands, so if you have a question, I think Sandra or somebody else can let me know. So let me now focus on the meat of the talk, which is doing pandemic science in real time. There's a beautiful article by Feinberg and Wilson in Science when 2009 H1N1 pandemic was unfolding. And I've been influenced by this article. I've essentially shown this article. It's an editorial article. And they talked about what aspects of real-time pandemic science response efforts are important. So, encourage you all to take a look at it. But the way we have participated in this is to build decision support environments for our sponsors. And we have actually been developing models to respond to pandemics and epidemic outbreaks since about 2002 and participate in every large outbreak that has happened since then. We started with a study for smallpox that was published in Nature, and I'll say a little bit more about that, to H5N1 planning. We talk about H5N1 today, but there was actually an H5N1 scare back in 2005, 2006 too. Some of you might remember that. And that led to a national influenza pandemic plan from the White House. After that, H1N1, then there was SARS in the middle, followed by Ebola, and then there was Zika outbreak, if you all recall, and then, of course, the biggest of all that we have seen, at least in our lifetimes, which is COVID. But the idea was that we wanted to build these tools not just for forecasting. There's often a lot of stress on forecasting, and that's one part of it. But I think models can be used to do a number of different things, like synthesize information, provide a range of interpretations, evaluate the range of responses, coordinate activities between stakeholders, and make usable systems that can be used not just by computing experts. So on the right, you'll see what a typical epidemic curve looks like. For those of you who have not studied epidemics, the curve basically shows the number of cases on the y-axis and time on the x-axis. And these are called phases of a pandemic. You start with very early cases like H5N1 is in the green side. It has zoonosis, which is the jump of the disease into humans for a sustained transmission. We are not there yet, luckily, in H5N1. We do get jumps, but largely stayed within animal kingdom. Then you have widespread human infection, followed by changes and then response, and then the pandemic goes down. That's typically how people are taught about pandemics in classrooms, right? But the interesting part is COVID pandemic, which all of you experienced, looked very, very different. In fact, I would say that it was a union of at least 4 different pandemics, if you want to use a single epidemic curve as a basis. So you had the first wave in April, followed by another wave in January 2021, followed by a wave in October, which was a Delta wave in 2022 or so, and then there's Omicron wave. Each wave, interestingly, produced very, very different spatiotemporal cases in the US. And the map on the top, just the colors should tell you how the prevalence curves look like during that time. And the ground conditions were different. In the first wave, it was a wild type. There was no immunity in the population. Mixing was severely controlled after the first wave started, extreme lockdowns. The second wave had relaxed social interventions. We had partial openings. People started to do behavioral adaptations. Mask wearing became more prominent. Partial vaccination started happening in January, if you recall. Delta was a really bad variant that came around, but again, we had different ground conditions. So each of them almost looks like a different pandemic in a certain sense. And that's what made response very, very complicated during this. So people might argue, why weren't we told early on to do certain things? The challenge is that the world kept changing on us in a certain sense. And that's what we expect in a big pandemic. So just to give you a sense of how our modeling group worked on this, this is before the pandemic started, we were monitoring the disease back in 2020. DOD actually told us they see something unusual in Far East. We don't know whether it will become a pandemic or not. We shut down the universities and other places in March. And then, as this timeline shows, we were able to participate at different levels through the pandemic response phase and do different sort of analysis. And I'll say a little bit more about it. Actually, here is a slide that is worth showing. So back in March 2020, which is now almost 5 years ago, to almost 5 years to date. DoD called on us, and we talked with CDC as well, and we talked to the White House pandemic planning group, and we were asked to constitute a team to respond to the pandemic. We worked very closely with those agencies, but in early April, we started working with our state as well. And from there on, essentially for 5 years, we have a group of 80 of us, and the number is smaller now, and largely now by April, we have started disbanding the group, but worked on this essentially every week for 5 years. And for a large period, people did not take time off. People were working full time, 7 days a week in responding to this. And I'll say more on the kinds of things we ended up doing. I feel very proud of the team. I feel very proud of being part of the team, because people really, did everything they could and set aside their personal interests and constraints to make sure that they did their best in doing this. And we had to do this over Zoom, if you recall. Most of us couldn't come. A few of us used to go to office, stay in our rooms and work. And it's very complicated time to work. We, of course, did a lot of work to support policy making, but we also published. We published a lot. We published more than 120 papers by now on this topic, just in the last 5 years. So I'm happy to share those papers. So here are the kinds of things we ended up doing. As you all know, we were the lead analytical group for our state. So if you visited VDH website, the data that you saw there was produced through the models that we had built out here. We supported VCU and James Madison University and some and parts of UVA's decision-making process. We supported CDC's hubs, which I'll say a little bit more, and then DoD, and also internationally in India and European CDC. So we built out a number of different tools. One of the early tools we built was to understand the COVID-19 dashboard. JHU dashboard then became more popular, and then our dashboard continued to be used, but not at the level that their dashboard got used, but was done at the same time. We did medical resource dashboard. There's a work on mobility-based analysis, and another decision support system for the state to understand the role of various social interventions to control the pandemic. We also supported Department of Health, and this is where I want to highlight the team effort. Brian Lewis took it upon himself to present the work on behalf of the team every week. And Andrew Warren, and then later on Anuruddha and Gursharan, all of them presented. But there was a time in the early phase that we used to have about 2 to 3 presentations to the health department here. One at the governor's office level, where Secretary of Health and Education Secretary and Homeland Security Secretary would participate. There was another group that was VDH analyst, and a third group, which was a task force that the governor's office had set up that wanted updates on how things were going on. And we did a fair bit of work with them, essentially close to 400 media and references in this, but the number 120 is actually now close to 200 updates. And all these slides are available on the web. So Brian essentially was speaking every week from March 2020 till about March of 2025, every week, literally. Remarkable work. And he was representing the group, right? I mean, he was not doing all the work, but he had to synthesize it and present it on our behalf. It was challenging, as you can imagine, and led to some very interesting results. One which I think has had direct financial impact was one where we told them not to build field hospitals, and has saved state close to at least $60 million. And the numbers they've given us, and this is published in the newspapers as well, is between $60 and $100 million by not building these field hospitals. And this was very early on that we engaged the state. And the question we were asked is, If you recall, New York was having a large outbreak, and they had built out. They're bringing in this Navy ship. I think it was called Mercy. That was supposed to be a floating field hospital. Javits Center was turned into a field hospital, and they were concerned that Washington is going to be the next place where this is going to happen. So we did modeling. We told them we don't think that we'll have that big an outbreak, and the group in our state took our word, and were convinced that that's the case. So they asked us a couple of important questions. They said, when do you think you can tell us if the outbreak will be large enough in our state? So we felt our models could never be good enough to predict beyond 3 to 5 weeks period. So we told them the best we can do is about 5 weeks. Luckily, the National Guard had the system ready so that they felt that within 6 weeks of our notice, they could build out a field hospital. So it worked out very well. So if you look at Brian's slides, all the projections are about 5 weeks forward and never more, because we knew that the ground conditions would keep changing over time. The second part that we told them is, to the extent you want to build field hospitals, you should build them close to medical centers, because it's not just the hospitals, but the personnel, the equipment, that is needed, and it's very hard to do it in a different place. The third thing we told them, which turned out to be useful, was we said, to the extent you want to build one, you should build what we call mobile centers, because pandemic moves. So today it might be that Richmond is not doing well, and maybe in 3 months from then, it might be that Charlottesville has a bigger outbreak, and then it might be Northern Virginia. So you should have the ability to wrap that hospital up and move it to some other place. We had learned this from our experience through the Ebola outbreak in Liberia, when we worked with the DoD, and turned out to be a useful strategy. So just to let you know that this is the kind of analysis that we could do with them very early on and had tangible impact, and that's how we started working with the state very closely. There's a report that the General Assembly was given. We also worked a scenario modeling hub at CDC that consumed this and was briefed to the pandemic response team at the White House. There are 19 different rounds. There are 5 different teams initially, and later on now 18 different teams that work on counterfactual questions. And our team has 2 different models as a part of this. There's really interesting work that is being done nationally to bring in multiple views on possible questions. The questions about primary vaccinations and NPI decisions. There was a question about vaccinating 5 to 11-year-olds. Very early paper that was published in MMWR on modeling the future of COVID-19 cases based on this. We also did mobility analysis. It was a nice small piece of work that we did with Stanford group. We used SafeGraph data and census data to help our state solve an important question about placing mobile vaccine sites. If you all recall, vaccines were in short supply before, and afterwards, when we had reasonable number, the question was, how can we distribute? We can't, vaccine hesitancy was not something we could change, but what we could make it is accessibility could be improved. So people who wanted it could have the vaccine. So we were able to use real-time mobility feeds from SafeGraph and other such apps to show them where these mobile centers could be placed so that folks would have easy access to the distribution sites and can get it. We also built another dashboard for the state on evaluating social interventions based again on work with Stanford. So let me quickly tell you how we approached this problem now that I've told you what we did. And this is a 3-step process, simplified a lot here. We first synthesize what we call a digital twin or digital similar of the place we want to study. We then build agent-based simulations on top of these, and then build AI workflows to study problems related to calibration, forecasting, explanations, and such. So what is digital similar? It is really a virtual representation of an area that we concern ourselves and is motivated by the problem we study. So for pandemics or epidemic outbreaks, A digital similar is a social contact network, patterns of how people interact with each other in a city. So it needs information on census, it needs information about built infrastructure, households and household forms, where people go and do their work, where do they travel. All this information is put together in a fairly complicated set of algorithms to build a digital twin. We started this back in our Nature paper in 2005. Stephen was the lead author on that paper. And we studied smallpox as an important question, and whether vaccines could be needed to protect the military and others. Now we can build such a twin. So that was done just for Portland. We've now come to a stage after literally 20 years where we have a digital twin for the entire US. So 300 million nodes in this network and about 15 billion in edges, which are time dependent. It's a really large network. So people who are interested in large scale problems, this is a beautiful network that we have created. And there's no final answer to this, right? As we get more data, we keep improving the network and modifying it. So in the last round, we have added information on agriculture workers. The round before that, and I'll tell you why we did that for H5N1 effectively. Before that, we added information about race and ethnicity because we found that COVID-19 had affected the populations, different segments of the society were affected differentially. And so we wanted to understand that. So we keep on adding new layers to this. What is interesting about this network is it has got all these different scales, right? The scale of a single individual, a household, a community, a region, and then the whole state itself and the country. There is no other way but to build this bottom up. And today we would use the term generative AI, but really it's a set of algorithms that are put together to build such a network. We've also expanded on this for our work on zoonosis and larger outbreaks, and we've created coarser versions of it for now large parts of the world itself. We've probably covered 70 to 80% of the world now. China is an exception. We don't have as much data, and then some other places don't have as much data, but most places now have been covered quite well by our data. We then build live twins. We take into account real data that is about epidemics, for instance, case numbers, serology, surveillance numbers, disease models, geography, vaccine schedules to understand how the twin evolves. We then run large-scale parallel simulations. For those of you who are computer scientists or have interest in it, these are extremely large simulations. You can imagine 300 million nodes running for 3 to 6 months of the pandemic is a non-trivial computing effort. We were lucky enough that UVA provide us very good access to the Rivana machines here, and we essentially consumed Rivana in large amounts for the last 4 or 5 years. But NSF provide us cycles at the Purdue Supercomputing Center and the Pittsburgh Supercomputing Center as a part of the national initiative on this topic. So one of the technical challenges we were able to overcome is that a single machine was never enough to do these runs. Even a large parallel machine was not enough. So we had to essentially come up with a new way to architect this so that the 3 machines, the Pittsburgh machine, the Purdue machine, and Rivana here, could be essentially pulled together to solve the problem that we had. So each of them was large, but each of them had constraints on it in terms of size, in terms of when you could run these computations. So what we really created is a virtual machine which had all the 3 machines embedded in it. And that was a really interesting computer science problem that we were able to work on, effectively creating a virtual machine of sorts. We worked on, as I said, various problems. I've already discussed some of them, so I'll jump through this. I want to mention some of the ongoing work, and then I can stop. I think this might be a good time to then take questions. A piece of research that we've done over the last now 9 months, which I think might be of interest to some of you here, is to extend our concept of digital twins or digital similars to represent livestock in the US. And we're doing this explicitly as a part of the CDC PGCOE project that Amy and I work closely on with other collaborators. Amy is the UVA lead. And the idea was that we want to understand the risk of infection in livestock and the possibility of zoonosis in the US through livestock to humans, right? So this is a very, very interesting data structure. It has got 4 different layers to this. We have, first of all, livestock, which is farm level information across all of US. And it has got multiple types. You have hogs, you have cows and cattle of different kinds in great detail represented in this. We have processing centers, milk processing centers, meat processing centers, poultry, eggs processing centers, because you know that some of the H5N1 transmission is happening through milk and working with those processing centers. Then we have the human population layer, because we need that for the zoonosis part, and then the wild birds, which is important. The wild bird is taken from the eBirds data set that Cornell and others produce. We used the data set and synthesized it. But effectively, these 4 data sets were brought together and into a very, very interesting data structure, with which we have now finished the first paper, but it's providing the CDC and USDA a risk map of which counties have a high risk of H5N1. And our results seem to do quite well retrospectively. So we are updating this essentially every month now to provide the federal government guidance in terms of surveillance, where they might look for. So this paper was just submitted. We are now also working on what's called conditional risk. Given that we have done some observations, where do we think the disease is going to move forward? We're also starting to do experimental analysis using our computer simulations of questions like, if the virus jumps into humans, what is the likely course that the virus is going to take? And the reason this is interesting is COVID came to us through Far East and came to the coast. It's possible that this particular virus might start its journey through rural America, right? Because that's where the interaction between humans and animal and livestock is much higher, and will form very different seeding patterns for this disease to start. So we're starting to work on that as well. So it's a really interesting data set. We have put it out in the open. You're welcome to use it. The other thing we are doing is trying to work with wastewater data, because we found that that's an important data set. We used it during COVID, in the second half of COVID. Now we are helping our state and CST with flu forecasting with it, but we can also use it for H5N1 analysis, because this provides a fairly good way to understand where the disease might be and what the rough prevalence might be. And we have used that to essentially build a forecasting tool that is in operation right now, which combines many other data sets, but also wastewater data to understand burden of a disease. We have been focusing for the last 6 months on flu. Before that was COVID. We can also talk about other diseases as they go along. But this data source is rather interesting, and this forecasting tool that we have built out, that is part of the CDC forecasting tool, is very useful. And this picture shows that actually the wastewater improves the overall forecasting ability that we have. So let me summarize, and then we can take questions. As I said, we have been working on pandemic response for 25 years now, and worked on almost all large outbreaks. We developed new capabilities in doing this. The part that COVID really forced us to work on is real-time pandemic response. We've not done it as much before. Largely it was planning style exercises, but real-time aspect was really interesting. And all of this has led to developing technologies for future outbreaks. We think that the technologies we created now will be useful in the next round, especially, vector diseases are becoming important for US again, with temperature changes that are likely to happen. H5N1 is certainly something that everybody's tracking. Hopefully it never materializes into a full-fledged zoonosis, but you never know. and other outbreaks that we're likely to see in the near future. The tagline we had when we had written our proposal is that the next pandemic is going to happen. The question is not if, but when. And I think that is still true even today after COVID-19. So let me stop by saying the following. I think what we are after in this round, which might be of interest for iThrive as well, is this concept of P-cubed analytics. We want to develop tools that have the three characteristics that we feel are very important. One is these tools are pervasive. They should allow decision makers to make decisions at any place, any time, on any device. This is a vision that Mark Weiser had for computer science, pervasive access to decision-making tools. It should be personalized, which means that it reflects the constraint that that particular decision maker had. A mother and father are also decision makers, but their purview is whether to go to work, whether to send their children to work, to school, whether to wear masks. A governor or a public health official has a very different set of decision making purview. So that has to be personalized for their needs. And finally, it should be precise in the time, space, and context that they're dealing with. You might be the mayor of Charlottesville, and your context is largely Charlottesville. You might be the governor of Virginia, and your context is the whole state itself. And you might be the president, and in which case, your context is the entire country. So with that, I'll stop. I want to thank you all once more for the kind invitation. I hope this was useful. I'm happy to take questions.